So we're going to start by looking at cell structure. We're going to be looking at eukaryotes and prokaryotes. So eukaryotic cells, which you should know from GCSE, are cells with membrane-bound organelles. And we'll look more at those in a minute. And we're talking here about your animals and the plants. So, if we first of all look at the structure of an animal cell, I draw a rough picture. So starting from the outside, we have the cell surface membrane. I've called it the cell surface membrane to distinguish it from other membranes within the cell. You can also call it the plasma membrane. And we should know from GCSE that it controls what enters and leaves the cell. Then within the center of the cell, we have the structures that make up the nucleus. So we have the nuclear pore, the nuclear membrane, in between which there are nuclear pores. Nuclear membrane again controls what goes in and out of the nucleus. Substances like RNA, can leave via the nuclear pore. Within the center of the nucleus, you have the nucleolus, which is a dense area, which is responsible for making ribosomes. And then that whole thing together, so that's the membrane and the pores and the nucleolus and the nucleoplasm inside, that all together is what we call the nucleus. Obviously, the nucleus contains DNA. It's the site of transcription, the first stage of protein synthesis, contains all the genetic information. If we keep on going around our cell, coming off the nuclear membrane as a continuation of that nuclear membrane is a network of membranes. Now that network of membranes can be one of two types, and they go all the way around the cell. Obviously, I'm only going to draw a few of them in. They could be the type that contains on them structures which are called ribosomes. And in that particular case, with ribosomes on it, this is called the RER, stands for the rough endoplasmic reticulum. And it's the site of translation, the second stage of protein synthesis. And that's because the ribosomes themselves, which can also sometimes be free within the cell, they are the site of translation. The membranes that do not have the ribosomes on, they are called the smooth endoplasmic reticulum, or the SER, and they are responsible for lipid synthesis. So they make all the lipids that are going to be needed in the cell, and as you will come across, lots of lipids needed in the cell surface membrane for the phospholipids and all the other membranes within the cell, plus other lipids may be required as well. Keep going into the cell membrane, we then have a very important structure which looks like a series of plates stacked on top of each other, a series of flattened discs. It's called the Golgi Body or Golgi apparatus, it's the same thing. And it's responsible for modifying and packaging proteins. So proteins will come from the RER in little vesicles, which are little tiny envelope circles, and they'll be taken, so I can draw a few in, they'll be taken to the Golgi body, like so, and the Golgi body will then modify the proteins and package them again into little vesicles to be taken out of the cell. So it's part of the whole sort of protein synthesis function of the cell. You also have within the cell very important organelles that you've come across at GCSE, which are double membrane structural organelles, and they are our mitochondria. Their job is obviously to release energy. Note, I note the word release, not produce, because energy cannot be produced in aerobic, specifically aerobic respiration. We'll look more at mitochondria in a little bit. They're very interesting organelles. And we also have within the cell these tiny little envelopes packed full, and they are known as lysosomes. And they're packed full of hydrolytic enzymes. What are hydrolytic enzymes? Hydrolytic enzymes are enzymes that can carry out hydrolysis, so they can break things apart. Remember, we always use the term hydrolysis. So cells will use them to digest old organelles, or in the case of phagocytes, to digest the pathogens that they've taken in. They'll pour on the hydrolytic enzymes. And then you also have one final structure. I mean, there are others, but one final structure we're going to look at, which I'm very simply going to draw like that. You're not going to come across it for a while. It's called the centriole. And it's responsible for making the spindle in mitosis. So if we move on down, and now we think about a plant cell. Plant cell has all of the same organelles as an animal cell, but some more as well. Okay, the only exception is that it doesn't really have is the centrioles. They're not found in our higher plants. So far, I've just again drawn in the outside of the cell. I'm just drawing in the cell surface membrane now, which again has the function of controlling what goes in and out. But outside of that membrane is our cellulose cell wall, so made of a polysaccharide cellulose, which, if you remember, we talked about was a polymer of beta glucose. We then obviously have our nucleus. I'm not going to label everything again, with our nucleolus and our nuclear membrane and nuclear pore. We still have our rough endoplasmic reticulum. We still have our smooth endoplasmic reticulum. Just put some ribosomes on here, like that. We still have some mitochondria. We still have our lysosomes. We still have our Golgi body. But what we do have extra in this cell is we have a very large vacuole, which may actually sometimes squash the nucleus into the corner of the cell. And the vacuole contains cell sap. It helps to control osmosis by controlling what it stores inside that sap. And then the really important organelles, which we really can't forget, they're much bigger than mitochondria. In their double membrane, I'm only going to draw one, which is our chloroplasts. And our chloroplasts are obviously our site of photosynthesis. They're really large organelles, much bigger than mitochondria. When you're looking at micrographs, in other words, TEM pictures of animal cells and plant cells, so look in the plant cells for the presence of the chloroplast, which will be bigger than mitochondria. You can tell the difference between a plant cell and an animal cell by the presence, obviously, of the cell chloroplast and of a cell wall. Remembering that there will be some plant cells that won't have chloroplasts, like root hair cells, for example. Now, I said I'd come back to mitochondria, and I would come back to chloroplasts. Let's draw a mitochondrion in. I'm not going to do it in a lot of detail, but here we go. So we have our outer membrane. and our inner membrane. And that inner membrane is folded, forming what we call cristae. But what I'm really interested in is the fact that inside we have some circular DNA, and we also have ribosomes. And they are sitting in the center of that mitochondria, which is known as the matrix. Now, if we look at a chloroplast, again, you'll come across these organelles in detail when you do the relevant parts. You've still got double membrane, inner membrane and an outer membrane. We don't call it the matrix, we call it the stroma. And sitting in there are these stacks, like that, in which you will find the chlorophyll. So stack is called a grana. Okay, and these are called thylakoids. But again, what am I interested in? I'm interested in the fact that again it has its own DNA and it has its own ribosomes. There is a theory that says that the mitochondria and the chloroplast were originally their own cells. They were prokaryotes of their own, and then they became incorporated into animal cells and plant cells because it was what we call a symbiotic relationship, which means it was beneficial to both. It's called the endosymbiotic theory. Okay, I said right at the beginning that eukaryotes contained membrane-bound organelles. So that's any organelles that have a membrane bound. Now, why am I going over this? Because we're about to look at prokaryotes, which do not. So membrane-bound organelles are your nucleus, your mitochondria, your RER, your SER, your lysosomes, Golgi Body, chloroplast. Those are your main ones that we're interested in. So let's have a look at prokaryotes. So prokaryotes are cells with no membrane-bound organelles. In other words, we are looking at the structure here of a bacteria. Now, different bacteria can have slightly different structures, but the basic principle is the same. So, once again, like before, we start off with a structure like that. We have a cell wall, but the cell wall is not made of cellulose, it's made of a substance called Peptidoglycan, which, if you can work it out from the name, must be a mixture of protein and carbohydrate. Once again, there is a cell surface membrane sitting on the inside. And it does have DNA. Its DNA is different. Its DNA is what we call circular DNA. So the DNA of prokaryotes is circular, whereas the DNA of eukaryotes is what we call linear. Okay, it's also what we call naked, which means it doesn't contain any proteins bound around it, whereas the DNA in a eukaryotic cell is wound around proteins. We then also have these extra circles of DNA, which you come across at GCSE, which we use in genetic engineering, which are called plasmids. They carry extra genetic information and they can be transferred between one bacterium and the next, often carry things like antibiotic resistance. And then they also have ribosomes, but their ribosomes are smaller than the eukaryotic ribosomes. And then sometimes you will also get, especially in particularly in aquatic Bacteria, you will get these structures coming out at the end, like looking like little tails. They are called flagella. Flagellum is one, flagella is two, and they are responsible for helping it to move. And then in some bacteria, but not all, you also get a slime capsule around the bacteria, which helps to protect them, for example, from acidity in the stomach. It's quite common for you to be asked to compare prokaryotes and eukaryotic cells. Okay, so if we think about prokaryotic and eukaryotic cells, now I could write no membrane-bound organelles, but then I'm going to get 1 mark. Whereas if I write no nucleus, no mitochondria, I might pick up 2 marks. I wouldn't go on to do that for all of the organelles, but do that for a few. We could talk about it having circular DNA, linear DNA, DNA with no proteins, what we call them histones, those are the names of the proteins, DNA with histones, small ribosomes, larger ribosomes, plasmid, no plasmid, flagella, no flagella. Important thing is that when you're comparing them, you compare them. So if you're asked to name 2 differences, you can't put on line 1, prokaryotes have a nucleus, and line 2, eukaryotes have mitochondria, because that's not a direct comparison. Line 1, prokaryotes have no nucleus, eukaryotics do. That's one difference. Line 2, prokaryotic have circular DNA, whereas eukaryotics have linear DNA. Marking point 2. Prokaryotes have smaller ribosomes. It's fine, because that's a comparative word. So you just need to be a little bit careful when you're actually comparing them directly, so you can look at exactly what you're talking about, make sure your comparisons are direct. Okay, this is going down in terms of what I might write. Be very careful again, because sometimes the question asks you how the structure of their DNA differs, not the location. So if I've asked how does the structure of DNA in prokaryotes differ from eukaryotes, I cannot talk about it not being in the nucleus. That is not structure. I would have to talk about no histones and it being circular and them having extra plasmids. So just be careful what the question's actually asking you about. So the last thing I want to talk about is how we study the cells, and that is the process of ultracentrifugation. Centrifuge is spinning. Ultracentrifugation, because we're going to spin it really fast. Also called cell fractionation. The same thing. Describes the same thing completely. So, first thing you're going to do is you're going to take your tissue, whatever cells that you're going to actually be extracting your organelles from. So it could be leaf, as an example. You're then going to add buffer. Now your buffer has to have 3 things. So the first thing is the idea that the pH is constant. The reason the pH wants to stay constant is to prevent proteins denaturing. It's got to be ice cold And that's to prevent your enzymes from functioning so that they don't break down the organelles. And it's got to be isotonic. Isotonic means that it is the same concentration as inside your organelles and inside your cells. And you've got to look at the question to decide whether you're talking about organelles or cells. So that no osmosis takes place, so that there's no bursting or shrinking. That was what could happen. So your buffer has to be ice cold and isotonic. So it's got three properties. It's a buffer to keep the pH constant. It's ice cold to prevent enzymes from working, and it's isotonic. You are then going to place it in a blender and basically turn it on. And that will break open the cells. Once you finish that, you're then going to need to filter it to remove any cell walls and debris. And then very simply, we're going to spin it. It's first going to spin it slowly, and that will remove the nucleus. And it becomes a pellet at the bottom. So if you imagine spinning it, what happens is you end up with a tube with a thing at the bottom of which is your nucleus, and then the rest of the liquid, which is called your supernatant, has all the other organelles in it. So you're then going to take the supernatant and spin it faster. This time the mitochondria, if you're dealing with an animal cell, or the chloroplast if you're dealing with a plant cell. So I'll put the chloroplast first. We talked about leaves at the beginning. And you basically keep repeating. So if you do it with an animal cell, first you get the nucleus, then you get the mitochondria, then you'll get things like the Golgi or the RER. It's very hard to say exactly which one. And eventually you end up with ribosomes. If you do it with a plant, first you'll get the nucleus, then you'll get the chloroplasts, then you'll get the mitochondria, and then you'll get the RER, Golgi, etc. And that's the end of something that allows us to take those organelles and do some experiments with them specifically.